Proteins are the worker molecules in living cells. They play an important role in virtually every cellular activity, including metabolism, cell division, apoptosis or programmed cell death, and cell-cell interactions. Therefore, it is not surprising that biological information that each gene encodes is about protein. Genes collaborate by forming gene regulatory networks, and their interactions are coordinated by external signals, such as hormones. Here I will explain a simple two-gene regulatory network where one gene activates another gene. We will see later that this simple network can serve as the basic building block for constructing more complex gene regulatory networks. Let's say we have gene X activates gene Y. So we have this notation X gene, arrow, Y gene. This simple notation involves, however, multiple steps. Let me first draw a DNA molecule here, and this is DNA. And we have Y gene here, which is part of this DNA molecule. And we also have promoter region for Y. And this promoter region is used to turn on or off Y gene. There are other parts of DNA that can be used to regulate gene activity besides promoter, but we will focus on promoter in this example. We have also X gene and promoter for X gene. Although we are talking about gene regulatory network or interaction between genes, genes cannot interact directly with each other. They interact through proteins that they make. I'm going to explain how this works. From X gene, X messenger RNA is made, and this process is called transcription. And from messenger RNA, protein is made. So we have X protein, and this process is called translation. And these two processes together is called a gene expression. And this X protein becomes activated, and I will show that activation using this symbol, asterisk symbol or star, in the presence of external signal, and let's call that signal S. And it can be a hormone such as insulin or growth hormone. Often this activation is achieved through what's called phosphorylation. This extra protein can bind to this promoter region of Y gene. In this case, this extra protein is transcription factor. It is an activating transcription factor because it activates Y gene and triggers gene expression. Now Y gene is transcribed and we have Y messenger RNA, and this is again a transcription process, and Y protein is made from Y messenger RNA. This process is again translation. Lastly, this Y protein is broken down. This is also a very important process, and we will discuss that later. Note that this X protein also goes through this degradation.